Hi. So you want to better understand nerves and the nervous system. So what we're going to do is we're going to look at some histology. We're going to look at some sections, some slides of nerves in a couple of different ways, and ganglia, so neuron cell body collections. I'm going to explain all of this. I think learning neuroanatomy is really difficult. We can look at nerves, we can look at the brain and the spinal cord, they're big physical things, but trying to understand how all this is wired up, what this really means, how it all works, I think is really, really difficult. You need quite an imagination to be able to imagine all these pathways. I think if we look at the cellular organizations, how the cells were arranged, how the tissues were arranged, show you what this really looks like on a microscopic scale, I think then it's easier to understand what a nerve really is, what neurons are actually doing, what we mean when we say ganglia or nucleus and that sort of thing. So we're just going to look at peripheral nerves today. I'll leave the central nervous system for another day, but this is going to be another one of those relaxed, live, slow, visual looks at these tissues and cells with descriptions of what we can see and what's going on without diving into too much detail, hopefully. Okay then, this is a piece of sciatic nerve. I don't know from which animal. So the sciatic nerve is the biggest nerve in the human body, right? It's a really, really big nerve. If you cut a cross section through it, you could still fit it onto a slide like this. But nerves are interesting because they're made up of really, really big cells, right? I mean, some neurons can be in us a meter long. So, you know, in really big animals, imagine how big they can be, right? Okay, so this is the lowest power I've got. And we can see lots of bubbles. We can see lots of circles. Okay. A little bit brighter maybe. So we can see lots of circles. This is the sciatic nerve. So this is, you know, we can see in green there, we can see connective tissue. It's mostly nervous tissue. There are going to be some blood vessels in there as well. It's like in many parts of the body we're seeing those four main tissues coming together, or at least three of them. So let me get, so here's a ruler, right? You know what a ruler looks like. Let me wedge that under there. It's not a pretty thing to do, but so those are the millimeter bits on a ruler. That's the sort of scale that we're working with here. And the reason I mention that is because the giant squid, not the giant squid axon, but the squid giant axon was one of the first neurons that was studied, and a single axon. So if you've got a neuron, you've got a cell body, and it sends off a very thin, it sends off an axon, sends off a very long process to another part of the body. So the job of the nerve then is to send a signal along that axon to a structure at a distance to make a muscle contract, for example. So squids do this. So squids evolved a method of triggering the muscles so they could, do that squidge of water to jet propel themselves away from danger rapidly. Now the giant squid axon was the first axon that was, squid giant axon. It's called a giant axon because it can be 0.5 millimeter in diameter, like the axon of one cell. So that process of an individual cell can be 0.5 millimeter in diameter. In fact, it can be 1.5 millimeters in diameter. So you see the scale that we're at here. So that would be a single axon. As I zoom in, you can see that big circle has got lots of little circles inside it. And if we zoom in again and give it a bit more light, you can see that we've got lots of individual circles in there. Now, we're looking at So we looked at blood vessels last week. So we're recognizing blood vessels there, right? Those circular tubes with space or bits of blood inside them. So now we're used to looking at blood vessels. But these little tiny circles, let me go to a higher power again and give it a bit more light. How's that? Right, so each one of these little circles, we're still not looking at the axon. There's a little dot in the center of that circle, that's the axon. So whereas a squid giant axon might be 0.5 millimeter in diameter or 1.5 millimeters in diameter, most of our axons, of our neurons, are less than 10 microns in diameter. So what's going on here is that the larger the diameter of the axon, the faster the propagation of action potentials along it. So the action potential describes the depolarization and repolarization of the cell membrane in one direction from A to B. It goes in one direction along the cell, and that's your it's at millivolts, but that's the signal that's conveyed along the axon to trigger something to happen at a distance from, say, the cell body. That's how we're sending signals around the body. What we've got, so squid are very old animals evolutionarily. Mammals and many other animals are a lot more recent. And these animals have evolved myelin sheaths. So what we're seeing in those circles, most of that circle is fat. There's a Schwann cell wrapped around the axon of the neuron. So the neuron is the central thread. And then most of that, say that pinky colour there, that is the Schwann cell with fat inside it wrapped around the axon. And that myelin, that fatty lining, it's like it's an insulator. So instead of the action potential having to run all the way along the membrane, it actually jumps between gaps. So where two Schwann cells meet, there's a gap called a node of Ranvier. And the action potential jumps between those nodes of Ranvier. And what's happened here is that whereas a squid giant axon might convey action potentials at, say, 25 meters per second in that huge neuron, these tiny neurons can convey action potentials at about 100 meters per second. So they're faster and they're smaller. Think about the eye, right? So the the retina, so the definition of your vision is restricted by how many neurons that detect light you can pack together in your retina. And those light detecting neurons have then got to send their axons back to the brain. If those axons were the same diameter as the squid giant axon, each optic nerve would be probably about the same size as your head. So it wouldn't work, right? So this gives obvious advantages. And if you think about dexterity as well, so these tiny muscles in the hand, there are lots more neurons per, say, square millimeter or cubic millimeter of muscle in the fine muscles of the hand than there are in the big muscles, because that gives us dexterous control. There are more neurons running to the fingertips, gives us greater sensitivity. So the more neurons you can pack in, the more things you can do, right? So it's very, very helpful. Now, you can maybe see that so we've got the green again, right? You've got your central thread of neuron, very difficult to make out, but you've got your central thread of neuron surrounded by your Schwann cell, and then there's a green circle around that. So this is, I think this is a Masson's trichrome stain. The green is connective tissue, it's collagen. So around the Schwann cell, you've got a thin layer of connective tissue. Type 1 collagen, I think type 3 as well. I've got some fibroblasts in there. You've got a little bit of connective tissue holding all of that together. And that's got a name. So that's the endoneurium. And then if I zoom back out again, if I go to a lower power objective, you can see this connective tissue ring here. So if I go to a lower power again, we can possibly see the whole thing. No, not quite. But you can see that we've got those neurons surrounded by endoneurium are bundled together by another layer of connective tissue. So this green layer of connective tissue we can see here, making that circle we saw at the beginning, that's perineurium. And then, I can't actually zoom out far enough, but then all of those bundles are bundled together with another layer of connective tissue, which is the epineurium. So you might come across those terms. Endoneurium is the smallest thing, and then the perineurium bundles those neurons and Schwann cells together. That gets called a fascicle. So fascicle is literally the Latin word that literally means bundle. So we've got those neurons and their Schwann cells. You bundle those together, hold them together with perineurium, and then you bundle those together with epineurium. And you can see that these are different sizes. A lot of the spaces here are artifactual. I think some of this is separated with sectioning and fixing and what have you. But we've got different size bundles. This is another then key idea about nerves, is that a peripheral nerve is a mixed nerve. It's carrying lots of fibres in here, and these fibres are doing different jobs. So we've got motor fibres that are going to be running to a muscle, but there will also be sensory nerves. Even if you've got a nerve that's only running to a muscle, you've got sensory information coming out of that muscle. So you've got these neurons are only doing, these neurons are only doing one thing. If you've got a motor neuron, it's only sending an action potential in one direction, away from the central nervous system and towards the muscle. And if you've got a sensory neuron, it's only sending action potentials in one direction, away from the sensor, back to the central nervous system. So in a peripheral nerve, we've got neurons that have a motor function. They go into skeletal muscle, and maybe we've got autonomic motor neurons in here that are going to smooth muscle, or glands, so sympathetic and parasympathetic fibres. And then we'll have sensory fibres in here as well. Like I say, if it's going to a muscle, that muscle will be sending sensory information back to the central nervous system, such as how long the muscle is, how stretched it is, nociception, pain, and that sort of thing. And there might be fibres in here from the skin, so all sorts of sensory fibres. Again, pain, temperature, touch, vibration, proprioceptive fibres from joints. So when we say that a nerve is a mixed nerve, this is what we're talking about. It's filled with all of these neurons, and these neurons are going to be doing different things, and some of them will be going to the same places, and some of them will be going to different places. So this is what we mean by a mixed nerve. Now, that section of the sciatic nerve was stained, like I say, with Masson's trichrome, which let us see a whole bunch of structures. This one, I think, is stained with osmium tetroxide. What this is doing is it's staining the fat black. So when we look at this, this should look similar but different. Look at that. Oh, wow, that's a really interesting image, isn't it? What on earth are we looking at here? Well, we're looking at a lot of fat, obviously, but let's find a nice one. We can see, so a lot of these cracks that we're seeing here are artificial there. They've been caused by, see, like that sort of shape there, these these cracks. These are artifacts of preparing the section and sectioning and that sort of thing. But that's a pretty good one. There we go. Look, let's zoom into this one. Where's it gone? Look at that. So the fat is stained black, and the fat is within the Schwann cell, wrapping around the axon. So now we've got a better idea of the scale and the size of these things that we're looking at, right? Sorry. Where's... Oh my word, and look at that. We're seeing lots of circles of different sizes, almost like these nerves are different, these neurons are different and doing slightly different jobs. And that's how a lot of neurons are classified, is by their diameter, by how much myelin they have. So the bigger the diameter of a neuron, the faster the action potential will be propagated along it. And also the distance between the nodes of Ranvier will affect how fast you send action potentials down it. So clearly we've got neurons here of different sizes wrapped with different amounts of myelin, so they're doing different jobs. Isn't that interesting? That's what we're talking about when we're talking about nerves being mixed neurons. It really jumps out at you there. Very cool. Now, I should go to a different plane of section if I'm going to see a node of Ranvier, because these Schwann cells don't, it's not continuous the whole length of the axon. There's a Schwann cell, little gap, Schwann cell, little gap, Schwann cell, little gap, and that action potential only needs to be generated at each node of Ranvier, hence that bum, 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 bum. Hence why it's so much faster than having to run the entire length of a neuron. But I'm kind of enjoying looking at this. Oh yeah, I had this nerve the other week, didn't I? Think we saw some in here. Let's have a look. Oh, yeah, okay. So we're looking at a peripheral nerve, but we're now looking at it in a longitudinal section rather than in a transverse section. So we're looking at the lengths of the axons, not cutting through the axons, which is why we're seeing long streaks instead of circles. So this again looks like it's stained in Masson's trichrome. So we got a lot of green because we're seeing that connective tissue. Oh, right, there we go, right there, bam. That was easy. So, what we're looking at there is we're looking at an axon running across the screen, and we're looking at the Schwann cell covering it, and we can see where one Schwann cell meets the next Schwann cell, and this has just caught the perfect plane of section so that we can see the node of Ranvier, the gap between those two Schwann cells, and that's where the cell membrane of the neuron will depolarize, and that action potential will jump to the next one. Now, because of the plane of section, there's no way if we follow this along we're going to see the next one, because it's just going to run out of the plane of section. Oh, but there's another one there. We could pretend that's the same neuron, but it's not. Can you see it? But we get an idea, don't we, if we run along the length. There's another one. That description of action potentials jumping from node of Ranvier to node of Ranvier to node of Ranvier is known as saltatory conduction. Are you getting a sense of how you've got all these individual cells bundled together, they're all running along next to each other, they're all conveying action potentials, they're all surrounded by Schwann cells. By the way, some neurons are not surrounded by Schwann cells. We just are paying attention to the ones that are. And then you've got that endoneurium, that little bit of connective tissue running around them. But that's your peripheral nerve. There's another node of Ranvier. But very, very cool. So the nuclei that we're seeing here, these will be the nuclei of the support cells, the Schwann cells and the fibroblasts, making the little bits of connective tissue around the outside. Got some very big nuclei. Okay, anyway, right. Now, because we were talking about the diameter of the axon and how that's important to the rate, the speed at which an action potential, a signal, moves along the axon. If you have a disease where you lose this myelin, where you lose these Schwann cells and these axons become demyelinated, they're unmyelinated, they're probably going to get to the point where they are unable to convey action potentials at all. It's not just that they slow down, although that could be part of it. It's likely that they will stop working. If you lose the myelin from these very, very small axons, these neurons will probably no longer be able to convey action potentials. And this is what we see in some neurological diseases. We see the loss of this myelin. Right, so this is us looking at the axons of neurons, and the axons of neurons come from the cell body. The cell body is somewhere else. So where is the cell body? Well, the cell bodies of neurons are much, much larger than their axons. You have your cell body, probably got a few little processes coming off it, dendrites, which are receiving inputs, and then it has that axon that may be very, very long, might be meters long or might be tiny, might be quite short, but that axon then runs off to somewhere else in the body, and that axon will end as a synapse, which will release neurotransmitters onto another structure and, for example, tell that muscle fibre to contract. But the cell bodies then, when we find the cell bodies of neurons, they tend to be grouped together because of the way they develop. So if the cell bodies are much bigger than the axons, you're going to get a bulge, you're going to get some sort of bulging gross anatomy. We see this throughout the body, dorsal root ganglia. In the case of the autonomic nervous system, the nice thing to point out are these guys here. So these are the sympathetic trunk. These are ganglia. So a ganglion is a collection of neuron cell bodies in the peripheral nervous system. If we had a collection of neuron cell bodies in the central nervous system, we would call it a nucleus. That rule gets abused, but you should call it a nucleus. So these are the ganglia. So this is what we're going to look at next, these collections of cell bodies, all right? All right, ooh, mucky. So this is a sympathetic ganglion. So this is what we just saw on the model. This is what you would find in the posterior abdominal wall. Don't know how you're doing with your autonomic nervous system layout, but okay. You have the first sympathetic neuron is in the spinal cord. So in the thoracic spinal cord, there's a lateral horn. That's where your preganglionic sympathetic neuron is. That's where you'll find the cell body. And then it sends an axon out, and that axon will travel here. It'll travel to one of the sympathetic ganglia. And then that preganglionic sympathetic neuron, its synapse is going to interact with the dendrites of a postganglionic sympathetic neuron, and that postganglionic sympathetic neuron will then carry the action potential on to the target structure. Maybe it's the smooth muscle in a blood vessel somewhere that's going to reduce blood flow through it and stop you from losing heat or something like that. But that's what we're looking at here. We are looking at the sympathetic ganglion, so we are looking at the postganglionic sympathetic neuron cell bodies. I know, it's a lot of words, but this is what we're talking about. So this is why it's making a lump, because these things are so much bigger than those tiny axons. Are you starting, might help if I brighten it up, right? Are you starting to see some of the cells in here? Again, we can see blood vessels. You can see why we did blood vessels last week, because we see them everywhere, right? Because they get everywhere. So we've got a lot of open circles there. We've got some artifacts again where things have come apart. But are you starting to see some big cells in there? How about now? So these are, the big circles. Most of the nuclei, the little nuclei that seem unattached, are support cells supporting the nervous system. But those big cells, you can see they've got a nucleolus inside them, a nucleus inside them. There's a circle, and they're inside the cell. I don't want to go to a higher power, do I? I think that's an oil lens. So inside the cell itself, it looks quite granular because neurons are quite active, they're quite busy, they have quite a lot of work to do. But that is the neuron cell body. So it's quite big. It's a lot bigger than the axon. I'm at what, 400 times magnification here. If we're lucky, we might be able to see some dendrites, but there's a couple of good neurons there. So these postganglionic neuron cell bodies, they will have little processes coming out of them, the dendrites, that will, like I say, receive inputs from those preganglionic sympathetic neurons that have come from the spinal cord. So if these are the cell bodies of the neurons that are running off as part of the peripheral nerves, these cell bodies might be called the perikaryon. You might come across that term, or just soma. Soma means body. But this is why we see those bulges. This is why we see ganglia throughout the body. The dorsal root ganglion, they're a little bit special, but most sensory neurons, their cell bodies will be found outside the central nervous system as a little bit of a bump. And that's why we've got a little bit of a bump, because we've got these cell bodies packed together, and you add up all those cell bodies, you get a little bit of a lump. It's bigger than the axons. The axons are tiny. They're tiny processes extending from here. But this is what a sympathetic ganglion looks like inside. All right. So we've looked at the neuron cell bodies, and then we've seen peripheral nerves, which are mostly axons of neurons from all sorts of places. Maybe their cell bodies are in the spinal cord, maybe their cell bodies are from a ganglion, maybe their cell bodies are from a nucleus in the central nervous system. And in the peripheral nervous system, we saw those axons surrounded by Schwann cells and other supportive cells, all bundled together with connective tissue. And that's what gives us a mixed nerve, a nerve doing lots of different jobs, running off to whatever part of the body needs those jobs doing. Anyway, now hopefully, what I'm hoping is that was a nice visual look. I showed you some real microscopic anatomy. So what I'm hoping is that when you're thinking about the anatomy of nerves, when you're thinking about the anatomy of the nervous system, you don't have to imagine quite as much. You've seen how it's really laid out. This is what the cells really look like. Hopefully then you can imagine how those cells are forming circuits and connections and sending those action potentials around the body and doing the things that they do. That was the aim here. Plus we chucked in a bit of terminology, all right? Well, I hope that was useful, and I hope it was interesting. See you next week.